Welcome to the Chemistry, Biology and Math Revision Hub. Today we'll be doing the Pearson Edexcel International A-Level Biology Unit 5 for January 2022. This paper is 90 marks. I will go straight to question 1. Question 1 says the photograph shows a transmission electron micrograph of a mitochondrion and the surrounding cytoplasm. So they say the mitochondrion shown in the diagram is 2000 nanometers in length. The length is, of course, that. So this is going to be 2000 nanometers. This is the real length. Okay, so they say calculate the width of this mitochondrion between X and Y. So what you have to do is you will use a ruler. You will measure this length from your image on the paper. That is the length. And then also measure that width using the paper with a ruler. When you measure, for me, when I measured, I got 24.5, which is going to be the length. And 75, the other is like that. I got that 24.5, and then the other is 75. So I divided and get this as the ratio, and then you have to multiply that ratio. And again, if one is, let me see, this is going to be the width, and the other is the length, you get a factor, and then you multiply by the other. So if we have, maybe it's going to be, this factor we're going to do when we multiply it with that, it's kind of like scaling up. If we have 2000, let's assume if the width divided by the length is equal, this is image, and then the other part is actual. This part is for the image, the other part is for the actual. So actual width, and then divide by the actual length. So it means when you divide the image width by the image length, or the actual width and the actual length, you have to get the same factor. So if I fit in my answers, 24.5 and 75 should equal to something I'm looking for, divided by the 2000 that I have been given. Of course, this is in nanometers. And then you're going to get 24.5 over 75 times 2000, giving you the value X. And as you can see, this is what I have here, because 24.5 divided by 75 gives you 0.33 times 2000, and then you end up getting 653. This is going to be in nanometers, as you can see here. I don't know if it makes sense. You're getting the actual and mapping onto the image, the actual and the image, and then you have to get the same answer. That is how it comes out. So I'll go to the next question. It says, describe how ATP is synthesized by oxidative phosphorylation. Here it means we use oxygen in the mitochondrion. One thing every answer requires is for you to state that ATP is synthesized through the chemiosmosis. So this earns you 1 mark. And then you say the electron carriers, of course, are going to deliver, or the carrier molecules will pass along the electrons to the electron transport chain. And as the electrons go through this electron transport chain, they release energy. This energy released will be used to actively pump protons from the inside into the intermembrane. So basically, they would pump it from the matrix into the intermembrane space. And then an electrochemical gradient is going to build up due to the increase of the protons in the intermembrane space. And now, due to the increase in the proton gradient, the electrons are going to move back into the matrix through the ATP synthase, and their movement causes the generation of ATP, as ADP is reacted with PI to produce ATP. The final step is when oxygen accepts the last electron as well as the proton. In this case, it is going to be the final proton acceptor. So you have to narrate the whole process, although I put more points than required, but at least it gives you the whole bigger picture of how chemiosmosis actually works. The next part says, describe how ATP is used to supply energy for biological processes. Using ATP, it has to be hydrolyzed or broken down by addition of water. So ATP is hydrolyzed and the bonds are broken. This breaking of the bonds releases energy, and that energy is what is used to run biological processes. I will go now to question 2. Question 2 says, muscles, bones, and joints enable movement of the skeleton. So part A says, which structure attaches one bone to another bone at a flexible joint? Of course, it's going to be a ligament. A synapse is found in the nervous system. A tendon does not do this. A tendon, of course, is going to be bone to muscle, and actin is found within the muscle, so it's not that. The answer should be B, which is a ligament. The next part says, how many of the statements about joints are correct? A, first part says, antagonistic pairs of muscles move bones at a joint. That is correct, because when one flexes, the other extends, or one is an extensor and the other is a flexor. That is correct. So muscles acting on a joint are myogenic. No. Myogenic, no. That is not true. And then tendons are more elastic than ligaments. Tendons are not that elastic in comparison to ligaments, so this is also wrong, leaving us with only one correct answer. Let's go to the next part. The next part says Usain Bolt's world record of 9.58 seconds for the 100-meter men's sprint is unbeatable. What is the role of myoglobin in muscle fibers? Remember, myoglobin is a form of protein. So A says it is an enzyme that reacts with myosin. No, myoglobin is not an enzyme. It is part of the sarcolemma involved in the release of calcium ions to act as an immediate source of energy to provide oxygen for muscles. Of course, myoglobin stores oxygen. Actually, it has a higher affinity for oxygen than hemoglobin. So myoglobin provides oxygen for the muscles in order to actively respire during activity. Part 2 says explain why the muscles of a sprinter have a higher percentage of fast-twitch fibers. Now we have two people. There is a sprinter and there is a marathoner. A person who runs a sprint, they require to run really fast within a shorter period of time. So they will need a big burst of energy, and this is why sprinters have a higher percentage of fast twitch, because they need fast reaction speeds. So I say sprinters run at fast speed, but for a short time. And since fast twitch fibers contract faster than slow twitch, of course, they are vital for sprinters. Also, fast twitch fibers basically carry out anaerobic respiration. So, when a sprinter is running, the oxygen they take in is not enough. So, there's a lot of anaerobic respiration going on, and the fast twitch muscle fibers are good for that. So, generating ATP in limited supply of oxygen, and this generates the energy for muscle contraction. So, fast twitch muscle fibers are going to be more in percentage when you are within muscles of a sprinter, basically. In an investigation, an athlete ran for 10 minutes on a running machine moving at constant speed. The investigation was repeated at 7 running speeds. At each speed, the heart rate and lactate concentration in the blood of the athlete were measured. The results are shown in the graph. So this graph is showing us the heart rate in beats per minute and then running machine. So we see lactate concentration as well as heart rate. We can see as the lactate concentration as the running machine speed increases, the lactate concentration increases, as well as there is a change in the heart rate. So let's see the other side. The first question says comment on the results of the investigation. Usually when you're commenting on such a question, you begin by looking at the bigger picture. The bigger picture is the heart rate as well as lactate concentration increase with increase in speed. As this speed increases, or as the independent variable is varied, you see that increases like that, and there is an increase in that as well. So this is the broader picture. And then you can go on to the individual ones. So I say the next part is lactate increases more. You can see this has a steeper increase than this one here. So lactate increases more at higher speed, while heart rate increases more at lower speed. So again, I want to take these ones away. This is the lower speed. The lower speed is, of course, in the beginning. We can see, of course, here lactate increases more at higher speed. At higher speed, you see there is a bigger incline. However, this one here, it has a slower incline at a higher speed. And then lastly, I say the relative change in lactate is greater than the relative change in heart rate. Of course, that's it. From the beginning to the end part for lactate, you can see that change is greater than this change here, than that change. So we can say this is a bigger change in comparison to the other. That is the third mark. You can be able to obtain his. If you want, you could also do a mathematical calculation to calculate the percentage change, or if you want to get the marks correctly. Let's continue to question 3. Question 3 says whales are large mammals. The photograph shows a beluga whale and a human diver. So basically that's it. So they say a beluga whale can become habituated to human divers. How many of the following statements about habituation are correct? Habituation is an example of anatomical adaptation. And then they say habituation is only observed in mammals. and habituation only occurs after repeated exposure to the stem stimuli. These are wrong, and that is correct. That is why there is only one that is correct. So B is the correct answer. I will go on to the next part. They say a beluga whale can respond to a stimulus using a spinal reflex arch. Which row is correct? So, location of relay neurons. Relay neurons are located in the gray matter of the spinal cord, whereas the location of the cell body of the sensory neuron is going to be in the middle of the axon. That is going to be correct. So part B is the answer here. And then continuing on, there is another part that says the heart rate of a whale changes when it is diving. The graph shows the changes in the heart rate that occur during dives of different durations. They say the maximum heart rate at the surface is when the whale returns to the surface after a dive. So let's see. Let's see the first part. So we have the heart rate on the vertical axis and the dive duration. So they asked you to comment on the changes in heart rate of the whale. Again, here you begin by looking at the bigger picture. Remember heart rate, that's in beats per minute, and then duration. So here I say the heart rate decreases when diving. As the diving duration increases, we can see the heart rate actually decreases. So I also say the longer the whale is diving, the lower the heart rate. Look at the distance. The longer it's diving, or the time, sorry, look at the time. The longer it's diving, the lower the heart rate. This is during the dive, and those are after the dive. So you can see during the dive, the longer it takes diving, the lower the heart rate. And then the next part, I say the difference between the maximum heart rate at the surface and the minimum heart rate during the dive increases as duration increases. What does that mean? Let me show you. It means this one and that. Let's see initially, let's assume there and there. And finally, we can see it's like it's diverging away. As the diving duration increases, the minimum heart rate during the dive and the maximum heart rate at the surface after the dive have the widest gap, or the biggest number, or basically the difference is going to be the greatest. I will go to the next part. So here, explain how the changes in heart rate are controlled when the whale returns to the surface immediately after the dive. This is about controlling the heart rate. Remember, when it's diving, it's using less oxygen. That is why the heart rate is low. However, it's going to produce a lot of lactic acid due to the anaerobic respiration that went on. So it means as it returns to the surface, its heart rate is going to increase in order to take up more oxygen to break down the lactate in order to remove the excess waste out of the body. So I said, during the dive, lactate in blood increases as blood oxygen decreases. And then, of course, the chemoreceptors in the aorta, this is the blood vessel, will detect the changes in the blood pH, and they will send impulses to the cardiovascular center, which is in the medulla. And that cardiovascular center will send impulses to the sinoatrial node, or SN, through the sympathetic nerve to increase the stimulation of the heart muscle. You guys know that SAN is found within the heart. It's the one that causes the stimulation of the heart muscle. So when you need more oxygen, or when your heart needs to beat more than the normal heartbeat, it means your brain has to stimulate the SAN to cause more stimulation of the heart. So this occurs, and then this will result into the heart rate increasing to maintain, so that it's going to increase and then maintain higher until the blood oxygen returns to the normal. And then the heart rate will be restored to the pre-diving rate it was at. This brings us to the end of question 3. Let's continue to question 4. Question 4. The kidneys perform important roles in filtering the blood and maintaining fluid levels in the body. Each kidney contains up to 1 million functioning units called nephrons. Each nephron contains over a filtering unit of tiny blood vessels called glomerulus that is attached to a tubule. The diagram shows part of the human kidney. Draw an arrow on the diagram to show the location of a single Bowman's capsule, which is the renal capsule. So, of course, the renal capsule is going to be located. You can see they've given us the cortex, so there is a cortex and there is a medulla. The Bowman's capsule is going to be located in the cortex and not in the medulla or anywhere else. So, around this area, wherever you would have drawn your arrow around this area, provided it was within the cortex, that it would be correct. I'll move on. So the next part says, where in the kidneys are most amino acids reabsorbed from the nephron? Of course, all amino acids are going to be absorbed in the proximal tubule or the first coil tubule. That is the correct one. And the next part says, which row shows the correct pathway taken by a water molecule through the kidney nephrons? The water molecule is going to move, of course, through the kidney tubules. It will begin with a glomerulus. Kidney tubules do not begin with a collecting duct. That's at the end. They do not begin with that, so glomerulus first. And then they go to the proximal before the distal, and finally to the collecting duct. So the answer here should be B. Then next, explain how the loop of Henle acts as a countercurrent multiplier to produce concentrated urine. Now, a countercurrent multiplier, things flow in opposite direction, and as you know, we have the loop of Henle like that. We have the one that is descending and the one that is ascending. So it's like things are flowing in opposite direction, and this is one way that makes it look like a countercurrent multiplier. And then in here, since sodium ions are actively transported out of the ascending limb, the water moves out of the descending limb. So the sodium ions are pumped out, and then water will move out of the descending limb due to the increase in the concentration created by the active pumping of the ions, basically, from the other ascending limb, basically. So I said, and the solutes from the ascending limb. So due to the increase in the concentration gradient between the tubular fluid and the interstitial space, so there is going to be increase in concentration around the one part of the limb, causing water to be drawn out, therefore concentrating the urine that remains inside. That leads to the production of concentrated urine within the kidneys, basically. Let's continue on. The next part says, describe how ADH, this is antidiuretic hormone, is involved in the control of water potential of the blood. Antidiuretic hormone is actually produced in the pituitary gland, and it will travel to the kidneys and cause the collecting ducts to be more permeable to water. So I said osmoreceptors. So again, they say describe how ADH is involved in the control of water potential. So it means we have to go back to how is ADH produced. Inside your brain, in the hypothalamus, there are receptors called osmoreceptors, and these monitor the water potential of your blood. So when they detect that there is a less water in your blood, they will send impulses to the pituitary gland, and this pituitary gland produces the antidiuretic hormone. So this hormone will be passed through your blood to the kidneys, where it will stimulate the collecting ducts as well as the distal tubules to increase the permeability of their cell membranes to water, and then more water will be reabsorbed from the urine, and this will the water potential of the blood goes back to normal. And then when it goes back to normal, the osmoreceptors will detect it, and fewer impulses will be sent to the pituitary gland, and then ADH will no longer be produced. That is how this works. So this brings us to question 4. Let's move on to question 5. Question 5 says, animals and plants can detect and respond to light. Rod cells respond to light by producing action potentials in the optic nerve. In which structure of are rod cells located? Rod cells are located in the retina. The retina is the light-sensitive part of the eye. It contains the rods as well as the cone cells. So they are located in the retina. And the next part they say, describe the role of rhodopsin in rod cells. Now, rhodopsin is a photoreceptor, of course, and when light is absorbed, the cisretinal rhodopsin, of course, the version is going to be in cisretinal version, and the cisretinal, as well as opsin, are converted into transretinal. So basically, it's cisretinal connected to opsin, and we call that rhodopsin. However, when light is absorbed, these go through a conformation change to change the part. The cis part is going to be converted into the trans part, transretinal. However, it's still attached to opsin. And then, however, when this conversion occurs, it leads to a cascade of reaction. Cascade of reaction is when one reaction leads into the other. So here I said it's converted that into transretinal and opsin, and this causes a cascade of reactions where the sodium ion channels close, And when they do close, the sodium is prevented from coming inside, and when it's prevented, the membrane now becomes hyperpolarized because, of course, the inside is going to be negatively charged in comparison to the outside. We call that hyperpolarization, and then this leads to formation of the generator potential. The cascade of reaction causes the sodium ion channels to close. Sodium ions are prevented from coming inside the membrane, and therefore the membrane is now going to be more negatively charged. We call that hyperpolarization, and that is when the generator potential is going to be produced. So the keywords you have to remember here are generator potential, hyperpolarization, that cascade of reaction, as well as the conversion between cis and trans, and that this is a photoreceptor. So let's move to the next part. The diagram shows the result of some experiments investigating phototropism. The diagram shows a coleoptile of a seedling. So this is the coleoptile, and then we see the roots. So there are various experiments carried out here. So the first one says there is an intact coleoptile illuminated from one side, and you see it bends, kind of as if it's responding towards light. Then there is another part where the tip of the coleoptile is removed and the remaining coleoptile illuminated from one side. However, here it doesn't bend, so it means whatever caused the bending was present in the tip that was cut off. The next part, they say the black cap placed on the tip of the coleoptile and the coleoptile illuminated from one side, so it did not bend. So it means even if you leave the tip there, however, when you cover it and expose light, there will be no bending. So that means there is something present in the tip, or in the tip of basically of the coleoptile, that responds to light and the bending occurs. So in experiment 2, we see 2A, they cut off, they say a plastic sheet is inserted between the tip of the coleoptile and the coleoptile illuminated from one side, and we see it remains upright. Yet here, when we put it on the other side, so they put it on this side, expose the light, the bending did not occur, but when we put it this side and expose to the light, the bending occurred. That means whatever that is causing the growth or the bending is going to be on this side. So it's like basically something is present at the top here that flows onto that direction, but not the other side. And when it flows onto this direction like this, it causes the bending when light is exposed to that. The next we see 2B, they say a gel layer placed between the tip of the coleoptile and the coleoptile illuminated from one side, and we see bending. Here we need to know that gel is permeable, so that whatever component is in here, of course this is a hormone, is going to go through, and of course it's going to cause the bending when light is exposed. Then the next part, the coleoptile is illuminated and the tip is removed. We see what happens. And then lastly, the tip placed on one side of the remaining coleoptile, that is then placed in the dark, and you see, of course, bending still occurs. So let's go to the questions. They say scientists concluded that these results suggest a plant growth substance is involved in regulating the phototropic response. Discuss how the results of these experiments support the role of plant growth substance in the phototropic response of plants, and use the information for these experiments and your own knowledge to support your answer. So here, of course, I have to go through all the experiments. Experiment 1, as you saw, it shows that causal phototropism is a chemical in nature, and it's produced at the tip. Because you saw when the tip is removed and light is illuminated, it does not bend. And then in experiment 2, we saw that the substance responsible for the phototropism passes through the gel, but not through the plastic. Again, I want to take you back slowly. We can see here, experiment 2, it does not pass through the plastic here, you can see, but it passes through the gel, and that shows that it is going to be permeable through the gel, and so that is why it caused the growth. And in experiment 3, we see that the chemical substance can diffuse and cause curvature of bending even in the dark. So it means once it goes through, whether there is light or not, there is a possibility of bending. So actually in the dark, it's not necessary that there is completely zero light, but there could be some wavelength that causes that to occur. So here I wanted to go through the chemical nature of the responses. Of course, you have to explain phototropism and so on. So I say phototropism is caused by auxins. Auxins are hormones, plant hormones. Phototropism, so it's caused by auxins. And phototropins like phytochromes, the ones you've seen previously, are receptor proteins, and these activate several hormones like auxins. So phototropins cause the hormones to be activated, so that some of them, like phototropism, basically the hormones are activated and then they cause certain changes to occur within this plant. So auxins are transported in the darker side than to the brighter side, and these auxins then cause elongation. So wherever they go to the other side, so if light is illuminated from this side, they go towards the darker side, and wherever they go, they cause increase of cell elongation, like you can see here. Elongation of the cell on one side, and this side they're not elongated as much, meaning it's going to bend like that due to overgrowth on one side. So the elongation causes the curvature due to cells being longer on one side than on the other side. And then always when they ask you these 6-mark questions, you have to give a detailed explanation of why this occurs. Here, I wanted to explain in relation to the cell membrane and how that basically the cell membrane is going to be, or the cell walls as well as the membrane are going to be changed in order to cause the elongation. So auxins stimulate decrease in pH of the cell, and this will activate the enzymes responsible for cell growth and elongation. So when these enzymes are, or even the auxins, will affect hydrogen ion pumps, this causes the intracellular space, of course the intracellular hydrogen ions, to increase. And when these hydrogen ions increase, the pH is going to be lowered. Lowering pH activates enzymes, because different enzymes operate in different pH ranges. So in this case, when the pH is lowered, the enzymes are activated to disrupt the hydrogen cross-links in the cell, and then, or in the cell wall in this case, and then this increases the osmotic gradient to the movement of solids in, and when the solids enter, it causes the swelling, and that swelling is kind of like growth or increase in size. You remember when cells are made, cells have to elongate by taking in water to push and cause the expansion or increase in size of the cell. So this process here causes water to come in, and when water comes in, it's going to cause the cell to increase in size, basically. This is all due to these auxins stimulating, causing a decrease in pH, and the decrease in pH activating the enzymes, and these enzymes would basically disrupt the hydrogen cross-links formed in the cell wall. And when this occurs, more water will come in, or basically water will come in causing the increase in size. This brings us to the end of question 5. Let's go to question 6. Question 6. Here we see the nervous system is a complex collection of neurons and specialized cells that transmit impulses between different parts of the body. Which substance can transmit a nerve impulse from one neuron to the next? So one neuron to the next, it's not going to be that, it's not going to be that, and it's not going to be that. Of course, this is a neurotransmitter, which is acetylcholine. That is correct. The diagram shows the change in the membrane potential as an action potential is transmitted in a motor neuron. So we see here the membrane potential as well as the time. So they say explain why the membrane potential changes between 1 and 2. So we see between 1 and 2, that is going to be, of course, depolarization. So let's see. I say this stimulates the change in the membrane. So stimulus, there is a stimulus that is going to initiate a change. Any change in the environment, in this case, it could be arrival of maybe a stimulation, and this could initiate change in the voltage of the membrane. When the voltage of the membrane is changed, the sodium ion channels are going to open, causing sodium ions to rush in, causing the membrane potential to, of course, be more positive or actually be less negative. And that, because of that, of course, we see an increase in the potential because it's less negative. And when this increase is greater than the threshold, then a depolarization generates the action potential. So you have to know the action potential, a threshold. As you know, this is an all or nothing law, so a threshold has to be reached, and then the depolarization, which leads to the action potential being generated. I'll go to the next part. So here we see myelinated neurons are surrounded by a fatty sheath. A graph shows the speed of conduction in myelinated neurons and non-myelinated neurons. So let's see, we have the speed of conduction on the y-axis and the neuron diameter on the x-axis. Then we have a straight line going through them. They say calculate the percentage difference in the gradient of myelinated and non-myelinated neurons. We are looking for the percentage difference in the gradient. So here I began by looking at the gradient of the myelinated neuron, as you can see, I used that point as well as that point and that point. So when I use that, it's 120 minus, of course, 60 over 20 minus 60, and I got 20 minus 10, and I got 6 as that gradient. And then this other one, because it's kind of a straight line, I use also, so we have this one, and we have the gradient of this one is given to us. So I say 6 minus 0.5 divided by 0.5 times 100, and that gives us 1,100%, which is the correct answer here. Remember, we are looking for the percentage difference. So 6 minus 0.5 over 0.5 times 100 gives us 1,100. Next part says, explain why the speed of conduction differs in myelinated and non-myelinated neurons. Now, you guys know presence of myelin kind of acts as an insulation. It insulates the neuron, basically, preventing basically it prevents the loss of potential or loss of electric signal from the neuron to the neighborhood, and this insulation creates the saltatory conduction, basically, which makes it faster. It's going to be fast because conduction will only occur at the nodes of Ranvier. So I said myelin covers the axon and acts as an insulator, and myelinated fibers have nodes of Ranvier where action potentials occur. So because of that, you see only conduction is going to occur at those points where we call the nodes of Ranvier. So we call that saltatory conduction will occur as nerve impulses jump from one node of Ranvier to the next node of Ranvier. So to get 3 marks, you have to include that myelin acts as an insulator, you have to include that there are nodes of Ranvier, and you have to include that saltatory conduction will occur through the nerve as impulses jump from one node of Ranvier to the next one. Let us continue. Here we see, they say, the positron emission tomography images shows the brain of an individual before control and after taking a drug, cocaine. So we see the normal one, we see the one who is a cocaine abuser, and another cocaine abuser. So cocaine abuser, day 10 and 100 days. So they say, explain how the positron emission tomography, PET, can be used to identify the changes in activity of the brain after taking this drug. A positron emission tomography uses a radioactive tracer which is added with glucose, and this tracer is injected into the bloodstream. When it goes into the bloodstream, it crosses the blood-brain barrier and it's carried into the brain, and actively respiring cells, of course, will need more glucose, and since this it's kind of behaving like glucose, cells that are actively respiring are going to take it up, and when they take it up, the sensor in the PET scanner is going to detect the radioactivity in different regions of the brain. So afterwards, a 2D or 3D image can be used to basically show the cells and see where or which cells are stimulated by the presence of cocaine. And then also the PET will show more regions taking up radioactivity in the cocaine, in the cocaine brain, basically. So an abuser of cocaine will have more regions taking up the cocaine, and that will be able to be detected, basically. This brings us to the end of question 6. Let's move on to question 7. Question 7, alpha-1 antitrypsin AAT deficiency is an inherited disease that can affect different organ systems. Alpha-1 antitrypsin AAT is a protein produced in the liver. So investigation of the breathing of a patient with AAT deficiency found that the resting tidal volume VT was 500 centimeters cubed, and the dead space DS was 150 centimeters cubed. The resting respiratory rate, RR, was 12 breaths per minute. They say calculate the volume of air that enters the alveoli, AV, in each hour. So they have given us the formula that we are going to use to do our calculation. So all we need to do is to get the values given here, convert them to appropriate units, and use them in the calculation. So AV, I would say AV is going to equal to, okay, let me first erase here. What I first did was to find VT minus DS. This is the part in that bracket. VT minus VS was 500 centimeters cubed minus 150 centimeters cubed, giving us 350 centimeters cubed. And that is breaths per. Basically, this is 350 centimeters cubed. That is the volume. So I multiply that by, because they said breaths per minute, so I multiply that by 12, and I got 4200 centimeters cubed per minute. So since I got this as the, of course, the heart rate, I had to convert that to the appropriate units. So if you want to convert that to decimeters cubed per hour, you will have to convert centimeters cubed to decimeters cubed by dividing that by 1,000. So 4200, when you divide that by 1,000, we will get 1,000. It's going to be 4.2 decimeters cubed. But remember, it's per minute, and 1 minute is 1/60 hour. So we come up with something like this. So 4.2 divided by 1/60, which gives us that. And therefore, the final answer will be 250 decimeters cubed per hour. That is the answer we are looking for. The key thing here was to know that you had to multiply by 12 here because they had 12 breaths per minute. That was the RR. So after getting this value here, VT minus DS, we had to multiply by the RR, which is 12, get the rate, which is 10 meters cubed per minute, and convert that to decimeter per minute and get our final answer. So the next part B says, activity of the heart was investigated in the same patient. The trace shows a normal electrocardiogram. So we see in a normal electrocardiogram, there is a PQRST wave. As you can see, P, there is a QRST wave. And so they said, name the stage of the cardiac cycle at T on this ECG. At T here, we have cardiac diastole or relaxation of the heart muscle. So basically, the heart muscle is in relaxation. So we call it cardiac diastole. I will go to the next part. So we have another ECG here. They say the ECG of a patient with AAT is shown in the trace. This is what we have. And you can see if you compare the previous one, let me take you back. You can see there is greater space here in comparison to the other one, which is kind of close. So it's kind of the rate is faster than the previous rate. So here they told us to calculate the mean heart rate. I'm going to use a different color because I already used red. Now, to find the mean heart rate, you remember they've given us that this is equal to 1 second. I want to take it away for you to see. So if you count how many boxes make 1 second, there is 1, 2, 3, 4, 5. So 5 boxes make 1 second. It means every small box is going to be, of course, 0.2 of a second. So to make my answer reliable, I used this gap here. You see this gap here. I checked the gap that corresponds to 2 complete beats. So let me remove so that you can see. I think you see this part here, and that part here corresponds to 2 heartbeats. So those are 2 beats. And the time here we have is going to be 1.2 seconds. If I can count for you, this is going to be 1 box, the second, the third, the fourth, the fifth, and the tiny part here which adds up to this to create the 6th box. So if 1 box is 0.2, 6 boxes are going to be 1.2 seconds in total time. So the rate I have is 2 beats in 1.2 seconds. So 1.2 seconds will give me 2 beats. And 1.2 seconds, if I convert that to minutes, I'll divide by 60. So 1.2 divided by 60 minutes will give me 2 beats. And therefore, 1 beat will give me 2 times 60 over 1.2, which gives me 100 beats in 1 minute. So the answer should be 100 BPM. That is the heart rate we can get. Here, I can show you the other explanation of how I did the other, but it's what I've actually explained to you. The next part says compare and contrast the ECG trace of this patient with the normal ECG trace. Now, when they say compare, it means they want similarities, and when they say contrast, they need differences. So we need to make sure when you see a question of compare and contrast, you know how to write the similarities as well as the differences. So the similarities between the two traces, all of them had the PQRST wave. And in both cases, the R wave was similar in height they had the same height of the R wave. The differences were in the person who had the condition, the gap between the T and the P was shorter, while meaning that the rate was faster. The normal ECG shows a lower heart rate. So in comparison to the two, the patient had a higher heart rate, and then the person who was okay, or the one who had a normal ECG, had a lower heart rate. I will go to the next part. The next part says one potential treatment for AAT deficiency uses adenovirus to genetically modify cells in an affected person. Adenoviruses contain double-stranded DNA. They say explain how adenoviruses can be used to treat AAT deficiency in a person. So here it's about introducing some genes. So we will identify the AAT gene. And when we identify this gene, we will use the same restriction endonucleases. These are restriction enzymes. They cut specific sections within the genes or within DNA. So we will use the same restriction endonucleases to cut the gene out of the gene of interest, as well from the to cut it out, or to cut the adenovirus DNA and insert this into the adenovirus using DNA ligase. So basically, we will cut out the specific gene of interest, and then we will introduce into the adenovirus. So the gene is introduced in the adenovirus using the DNA ligase. DNA ligase is used kind of like it's genetic glue. It seals the places where two sections of DNA are being combined. Then the adenoviruses can enter the host cell. So basically, this is going to be the vector. A vector delivers the genetic material into the host cell, and this will combine the genetic material with that of the host cell. So in the end, we're going to produce, the host cell will be able to produce a functional protein, which will be helpful in overcoming the condition this person is facing. This brings us to the end of question 7, and heading on to the scientific article. So for this section, again, I'm not going to read the whole article. However, I'm going to make you view it slowly. So this article, let's see, I'm going to just peruse through slowly so that you can be able to capture your screen. So there is that section. This is about food toxicology. I will just move up like that, and then I will go to the next page. We have paragraph 6, paragraph 7, there is paragraph 8, 9, we have 10, and then I'll move to the next page. There is 11, 12, 13, 14, 15, 16, moving on. The scientific document you have studied is adapted from an article in Science Direct, Assessment of Food Toxicology by Alexander Goslow. Use the information from the scientific article and your own knowledge to answer the following questions. They say explain how toxicants in food could cause Parkinson's disease. So it's in paragraph 3. I can take you a little bit back to paragraph 3, but I know we're not going to read everything. So let's see. Paragraph 3 from the scientific article says, the mechanisms of toxicant effects are multifactorial, interacting intrinsically and extrinsically with key molecules which play major roles in cell integrity, metabolism, signaling pathways, gene expression, and translation. For a variety of toxicants, their effects appear to converge on the generation of electrophilic species. These are very important, leading to oxidative stress, chronic inflammation. Oxidative reactions induced by toxicants lead to accumulation of damage. This is important. The toxicants lead to accumulation of damaged macromolecules, thus harming cells, tissues, and organs. So this is the very important part here in trying to answer the question, because they say, how are these toxicants going to lead to diseases like Parkinson's? And then they continue to say, therefore, toxicants must play central roles in the death of chronic inflammation, aging, and degenerative diseases such as Alzheimer's, Parkinson's, Huntington's disease, as well as multiple sclerosis, myocardial infarction, atherosclerosis, diabetes, rheumatoid arthritis, sterility, cataracts, and many others. So I will take you back to the answer that I wrote. So since they're asking us how to explain how toxicants in food can cause Parkinson's disease, of course, these toxicants are going to be absorbed in the gut, and when they go into the bloodstream, they'll cross the blood-brain barrier, and they will go into the brain and enter, of course, be fused into the neurons. So they will prevent the secretion of dopamine, and this will inhibit the uptake of dopamine by motor neurons because there will be no dopamine, or motor control centers of the brain, and this will lead to loss of muscle control, like the people who have Parkinson's disease actually experience. This one I meant motor control, not motor neurons. So the next part, they say suggest how genes affected by toxicants can be identified in paragraph 4. Paragraph 4, they talk about using microarray endonucleases. Again, I'm going to take you back briefly. Paragraph 4, I'm really sorry for this, but I have to do it. They say for in vitro assessment, a variety of biochemical systems have been developed to analyze damaging effects on integrity and activity of key biomolecules. Such molecules are important in cell integrity, metabolism, signaling pathways, as well as gene expression and translation. The list of affected molecules is extensive and includes enzyme receptors, membrane lipids, nucleic acids, or factors involved in gene expression. On a cellular level, a variety of viable assays. So this is what we are looking at, the assays that can be used. So a variety of viability assays are routinely used to quantify effects of potential food toxicants for the extrapolation of a range of dosages used in maximal tolerant concentration for the in vivo animal models and also clinical settings. So they say in vivo rodent models still appear to be a gold standard for toxicity assessment, but there are limitations such as traditional testing, such as high costs, low throughput readouts, inconsistent responses, ethical issues, and concerns of extra polarity to humans. So I will go back to the question. So because this is about analyzing the toxicants, we could use microarray to see or to identify the affected gene in the sample. And when we get that gene, we can cut it out using the endonuclease enzyme, and then we can stain and produce cDNA. I think you guys studied about this, using a fluorescent dye. We can also, after analyzing using the cDNA we've got and the fluorescent dye, we could also use bioinformatics to analyze the data produced from microarray and make possible conclusions or correlate the data to be able to understand how these come about, basically. The next part is saying, explain how scientists could determine the high degree of homology of genes between humans and zebrafish. If you want to determine the high degree of homology, meaning similarities between humans and zebrafish, this is always going to be DNA profiling. So we'll use PCR to amplify, and then we use gel electrophoresis to be able to compare the bands. So we compare the bands to show the similarities between the bands in the human as well as the bands from the zebrafish. And then the results will show that the product of the genes to have similarities. So any similarities will show that there is similarity between that of the humans as well as that of the zebrafish. So part D, they say, suggests how electrophilic species or free radicals could be involved in the modulation of gene expression. Again, in paragraph 6. Now, these electrophilic species or electrophilic species, we call the ES or free radicals, they can target and bind to transcription factors. Again, I don't have to read paragraph 6 because I showed you. The key thing here is they can bind onto transcription factors, and that is how they're going to modulate gene expression. So when they bind, they could cause they could also cause the shape of the transcription factor to change, and if it changes, then they can prevent it from binding to the specific DNA segment, and if that doesn't occur, RNA polymerization will not occur, meaning formation of messenger RNA may not occur, and if it does not occur, a specific protein will not be produced. So I will continue to the next question. The next question says, suggest how the ES, electrophilic species, can damage mtDNA, that is in paragraph 7. So in mtDNA, you guys know there are phosphodiester bonds. So these could break the phosphodiester bonds, and the strands of DNA will be broken up or will be disintegrated. And if the strands are separated they could also cause the breaking of the strands or separation of strands when the hydrogen bonds are broken. Another thing is they could cause mutations, and when these mutations occur, of course, there are going to be changes to the DNA, and it will no longer be the same. The next part says, if the MTT assay reduced NAD converts the MTT solution from yellow to purple, describe how the MTT assay could be used to show which of the two drugs is more toxic to the cells in paragraph 10. So we can use living cells, and then the reduced NAD produced, of course, during aerobic respiration. You guys know in aerobic respiration, NAD is reduced. And this reduced NAD can act as a carrier, leading to the production of ATP as electrons are carried by reduced NAD. So in this case, these reduced NAD produced from aerobic respiration, we can use the same concentration of the drug in doing so, and then we will record how long it takes the solution to turn purple When it does, the longer it takes to turn purple, then it means the more toxic the drug will be. Again, you have to go back and read paragraph 10 in order to be able to break down this answer. So let's go to the next part, which is the last part of the scientific article. They say, explain the advantage of reprogramming ordinary somatic. Somatic is about body cells, ordinary somatic cells to behave like embryonic stem cells for the use of, used basically in stem-based assays over toxicants. Converting normal body cell, ordinary body cells into embryonic stem cells could kind of have some issues of its own, because different people have different genetic makeup. So if you have individuals with different genetic makeup, the chances of rejection would be reduced. So in this case, if we could convert this somatic into stem cells, it means we have the possibility of making or of donating or helping so many people in these stem cell-based assays. And then we also know that different people could respond to different levels of toxicology or toxicity, basically. And so if we reprogram the cell, it will enable personalized response. So if somebody is sick, we get their own body cells and we convert them into stem cells, and then we will use those stem cells into therapy, meaning that person's immune response will not, the cells we introduce into their bodies will not face immune response, basically. They will be welcomed because they are from that same individual. And then this other thing will solve the ethical issues around stem cells, embryonic stem cells. So if we use the personalized cells to convert them into stem cells, then it means we no longer have to use embryonic stem cells in research, and the ethical issues surrounding that will be overcome. This brings us to the end of this paper, the January 2022 Unit 5 Biology for Pearson Edexcel. Thank you for being with us and hope to hear from you in the next video. Please do not forget to subscribe to our channel. Goodbye.